Thanks, Brian, for the highlights. So I recorded a video due to the instability of the servers this morning. So I think it might be better to play the video. And anyone feel free to stop me anytime for any question. And so let me play the video right now. Okay, so the previous version of tranSMART can only process the subject level data for statistical analysis. And the new version can store metadata, for example, the information files related to a program, a study, an essay, or a analysis, making tranSMART a all-in-one depository. And let me hold it for a second. And the browse is the interface to view, manage, and search those metadata. The Analyze tab, formerly the Dataset Explorer, is now synchronized with the Browse tab, meaning that they share the same selected studies, which is extremely useful because after a study of interest is found, user can switch directly to the Analyze for the analysis of the study. And there are 5 types of objects in the browse. They are the program, study, essay, analysis, and files, and file folders. The file folders can reside at any level. And so this is the node of a program. Clicking the node displays the information related to this program. Title, description, associate text, and associate studies under the program. Right now, there's only one study under this program. And the power users can also have the ability to adding studies or adding folders under the program. Clicking the study node displays the related information for the study. And similarly, the power users have the ability for addition of analysis essays or folders. And there is a analysis node under this study node, and the users can add new folders or delete essays under this node. And so there is a folder under the essay node, and the folders can be a place to save files. Here there are 3 files, and users can add or delete files. Notice that right now in the export cart, there's only there was no. Okay, let me go back a little bit. Okay. Notice that after user clicks the add to export, the files is added to the cart, and clicking the cart displays the file, and the user can download the file. So if there is a large number of studies, it might be difficult to find the study of interest. So the browse provided a search functionality. Now, if I type test, it would display all the nodes with the word in its name, or like in this node, in the description. Then users can use the filter to further narrow down the list. For example, if the user is interested in a cancer studies, notice that between the search categories, it's the and relationship. But within the categories, it's the or relationship, which can be changed to and as well. And finally, the list can be further filtered to only one study. Now let's clear all the filters. There are different types of fields in the searching, except the free text, which searches the node's name, description, and the file content. All the other fields have the auto-complete capabilities. For example, if I want to search a gene, I will select the gene field and type ERBB, and it will auto-complete the item. So I search the ERBB2, and here are all the studies that with this gene as a biomarker. And by clicking Open in Analyze view, it switches directly to the Analyze tab. Notice that the filter persists through the Analyze tab. And that's the basic functionality of Browse. We also enhanced the capability of tranSMART by developing the ability to take the new high-dimensional data types. The new data types are the mRNA-seq, microRNA-seq from NGS and qPCR, and proteomics data from protein mass spectrometry and immunoassay from RBM, and also the metabolomics data, which are the quantities of metabolites. Now let's see an example using the RBM data. Let's switch to Analyze tab and search RBM. And let's check the female as subset 1 and the male as subset 2. And under advanced workflow, let's choose hierarchical clustering. The clustering would be done on the RBM data of plasma. And let's click in the HDD button. Now let's use all the data. So if we do not input anything here, we would be using all the data. And now let's run. and it's taking a little while. So here is the heat map into subsets. Yeah, now we see the heat map and hierarchical clustering on the RBM data across all the samples. So since all the high-dimensional data types works the same way with the advanced workflow, I'm now going to demo more of the high-dimensional data type. The next one is the serial data. Serial data is a series data on the same sample, maybe at different time points. It could be either low-dimensional data or high-dimensional data. In this new version of tranSMART, each node of the serial data can be associated to a numerical value, and the serial data can be plotted along the scaled x-axis using line graph. And let's see an example. Let's select the study test serial clinical data, which contains the low-dimensional clinical data at different time points. And let's use the line graph. And let's check the glucose level at different day points as the time concepts. Let's remove day 14 so we can illustrate the point that those time points will be scaled on the x-axis according to its numerical value. And let's check the trial arms to the group concept and then run. Now we see that the x-axis are the different time points, and the day 7 and the day 21. They are scaled according to their numerical value. And the y-axis is the glucose level. So the different colors are 3 different groups. The serial data also works with the on the high-dimensional data. Now let's drag this serial gene expression data into subset 1. And again, select line graph. And let's check the baseline, week 1 and week 2 as the time concepts. And let's select the gene ERBB2. And let's drag the sex as a group concept and run. And now we see that the x-axis are the different time points, and the y-axis is the expression of the ERBB2 gene in the two groups, male and female. If we want the whole data set to be in one group, we can clear the group concept and just run. And yeah, so now all the samples are in one group. So the next example is the heat map of high-dimensional serial data. Let's clear this one and drag the female as subset 1 and male as subset 2. And under advanced workflow, let's choose heat map. And let's drag only the baseline and week 2 into the field, and let's select all the genes in the apoptosis pathway. And let's aggregate all the probes. Yeah, so here is the heat map. We can see that the columns within each subset are ordered by the time points. And we can also specify to group by subject instead of the time points within each subset. Now we see that the columns are ordered by subject instead of time points. So besides the pros, new HDD and the serial data, we also made a significant improvement on the grid view. Grid view is a great tool to get a quick sense of the data, but the previous function was pretty limited. And in this new improved version, the grid view can display sample IDs, display categorical variables in a single column. It can hide or show columns at user's choice, and the user can also export to Excel at the selected columns or all the columns. So here is an example. Let's drag the diabetes to subset 1 and the obese into subset 2. And let's run the summary statistics. And then let's view the grid view. And we can see the obese and diabetes, they are at different columns. And there is also a risk column that has nothing. Now if we drag the trial arm into the grid view, we can see that now the category variables, they are in the same column. And now we can delete those columns that we do not need. And we can also drag a numerical node into the grid view. yeah, now we see that the height is in a grid view. So the whole table can be exported to Excel, and users can also select specific rows to export. Yeah, so I think it's a extremely useful feature to get a quick sense of the data. The last improvement is on data export. When I recorded this demo, the server did not work, so I did not demo this. But basically, instead of export the whole data set, now the user has the option to drag the individual node into the data export field and to export only those nodes. So finally, this improvement is a team effort, and at Sanofi, the team members are Claire, Anik, and Sharon. In Genzyme, it's Sherry, Adam, and me. Our partners are Converge and also the Hive. Finally, I would like to thank the tranSMART Foundation for the great effort to make this community project possible.